Yeah. So everyone, we have so far seen module 2, and we have seen the useful features for biofuels. We have certain points remaining in the module 3, which are substrate digester and microorganisms in the process of biogas production, biomethanation, that we will cover today. Plus, we will cover rest of the things in detail, including production of bioethanol from sugar, molasses, starch, cellulosic material, and also we will cover the ethanol recovery, including some points on fermentation also. At the end, if the time allows, we will see the microbial production of hydrogen gas. We have already covered biodiesel, so we will briefly touch the points which are already covered. So we have covered what are the biofuels, including the definition of biofuels. As we know, it has to be 80% minimum content legally derived from living organisms harvested within the 10 years. That classifies as a biofuel. Uh, so it is a fuel consumed or produced from organic products and waste, particularly waste material, not food substances. Bioethanol is made from sugar, algae, wheat, and sugar beet, which are starchy sources, sugary plant products, and biodiesel is made up from oily sources like vegetable oil, algal lipids, and animal fats. Third component, which is a biomethane, which is we are going to see today for biogas, it can be produced from waste organic material, sewage, agriculture waste, and domestic waste. So for bioethanol, remember these are the sugary, starchy substances. Some are waste, some are other plant materials, but they are fresh. They are recently harvested, which can be used and dried in a powder form for bioethanol. For biodiesel, these are oily substances, again derived from certain oil extraction processes as a byproduct, like algal lipids, vegetable oil, and for animal fats, it is a byproduct of butcher industry. Except that biomethane is a product of all sewage, mostly, including agriculture organic waste material and domestic waste in animal waste. That is the fecal matter of the animals. So all this is a part of a carbon cycle. Whether you use it is in form of sugar, in form of as a source of methane, or in form of a source of uh, oils. So basically, it is a carbon which is playing role. We have seen this carbon cycle in previous lecture. There are three generations of biofuels as we have discussed in previous lecture. We are not going into detail. So we have covered these feedstocks also based on starch and oily substances. For biogas, we will use waste digestion end products like fecal matter mostly. So for today's lecture, we will see into detail the process of uh, alcohol fermentation. It starts from milling, that is cutting the raw material into pieces, followed by their fermentation and distillation and dehydration. So as we know from bioethanol, you need starchy materials. Uh, so starchy materials are generally encased in grain hulls. That means seed cover is there, and but the starch is readily available. So first you have to dehull it using in the process which is called as milling to export the starch contained in the seeds. And along with that, sometimes before milling or after milling, some the obtained seeds are moistened and allowed to sprout. So that allows conversion of starch, which is a polymer, into some oligomers. And then the sprouted seeds are dehydrated and powdered, and the powder is called as malt. So that process is similar to any alcoholic preparation of any alcoholic beverage like beer or like this. and the problem associated with it fine flour is difficult to remove in distillation, and so that we have to use the pot still that we will come in the next few slides that will reduce this problem. And followed by this, it's nothing but a preparation of mash, or which is called as cooking. Remaining starch is liquefied by boiling under pressure to dissolve water soluble starch. Basically, all malting, milling, and cooking is required to make hydrolysis of starch into certain oligomers, which can be acted by microorganisms to produce e- ethanol anaerobically. That's it. So cooking is done always under steam pressure at 100 degrees. All these things required for proper breakdown of starch, so that it can be, in a less time, it can be further degraded to form oligomers. Alternatively, you can perform milling and cleaning in single operation without the use of water, which is called as mash. And the next step to that is the conversion of those oligomers or fermentable sugars by certain enzymes into malt by acid hydrolysis. So generally nowadays enzyme extracts are used. So there are three types of enzymes. Alpha amylase produces dextrose or glucose, D-glucose. Beta amylases produce maltose, and glucoamylases reduce the remaining starch. That means more dimers, trimers, and tetramers are produced from oligomers and starch polymers. So in acid hydrolysis, as the name suggests, acid is used to break those bonds in between sugars, and the acid has to be neutralized before the further processes. It is done by calcium hydroxide. So this is done particularly in case of those particular polymers of sugars or starches which cannot be converted easily using this thing, microbial cultures and microorganisms. And at the end, pH is stage check. Cooling coils are used to cool it. Here, it is high temperature is required, acid proof equipment is required, mineral acid is used, and then followed by cooling. So, in the last stage of conversion of this fermentation, this is the last step of fermentation is conversion. It is glucose is add produced glucose through this cooking, milling, and malting is catalyzed into the glucose is processed into a pyruvic acid cycle or TCA cycle to produce pyruvate molecules, and further it goes anaerobically from the step of 2 acetaldehyde 2 molecules. So 2 molecules of acetaldehyde, from 2 molecules of acetaldehyde, 2 molecules of ethanol are produced under anaerobic fermentation. We know that after glycolysis, glucose produces two pyruvate molecules. That pathway is same for both anaerobic and aerobic. But from pyruvate, two ethanols are getting produced. So this is intermediate, which is called as acetaldehyde. From this acetaldehyde, during which you release two CO2. So in here you can see the oxygen going out of the system. So in along with that, later here it is getting reduced to form alcohol. Meanwhile, NAD and NADH are used. NAD plus is used as oxidizing or reducing agent like this one. Where it is used, ethanol is used as solvent, most of the things as chemical solvents, and it is for our interest, it is for fuel. Internal combustion engines, as we have seen in previous lecture, it has combined with petroleum generally, because to use absolute ethanol for engines, you have to use very good engine modifications, which is not possible. Which organisms are used for ethanol production? Mostly they are yeast, or there are many bacteria which can produce ethanol, and depending on the sources you can get different end products like other alcohols, butanol, isobutanol, or propanol, like that. Organic acids, you can get acetic acid, polyols, polyalcohols, and ketone gases are also produced. That depends on the culture which we select. Many bacteria metabolize sugar by EMP pathway. So instead of uh, glycolysis, pyruvate is not produced. The glucose is used using another EMP pathway, which is used by Clostridium, Sarcina, and Streptococcus. Mostly we have come across with yeast as why they are of interest, because they have high ethanol yield, high growth and fermentation yield, good osmotolerance. That is very important because you are using harsh conditions sometimes like acid, and you are neutralizing it at the end, but still they are good candidate organisms to produce ethanol due to these reasons, and they are very sturdy under physical and chemical stress. Coming to the raw materials of ethanol fermentation, mostly they are starch, cellulosic, and saccharine materials. So saccharine or sugar containing materials are glucose for fructose, maltose, 6 to 12 carbon sugars in fruits like these. So here we cannot use fruits directly, but you can use some remaining parts or thrown away parts of fruit, and you can use residual sugars from them if it is. Otherwise, the fruits which are gone beyond the step of eating, you can use them because they will have your simple sugars more as compared to starch. So over-ripened banana, for example, we can take here over-ripened banana, over-ripened grape. So over-ripened banana, you can't consume it for edible purposes. You can use it here. So it has to be over-ripened as an ideal case, because this will increase in over ripened, or what we call, which has gone ahead of eating. So they will have glucose, fructose, maltose more and starch less. Molasses are generally used in our country because they are readily available, and almost most of the time sugar factories are associated with ethanol production because of access to those molasses, as we have seen. Good point with respect to molasses is, if you compare the sugar content of fruits, you will find good sugar content in fermentable sugars, 52 to 55%. But the molasses above 15 to 20% sugar need to be diluted. So what is done first, the molasses are diluted by using water, and then they are allowed to be fermented. Cane sorghum means bajra in Hindi or bajri in Marathi. So leftover residues of this plant can be used because it has good starch content. Extraction requires heavy duty, but you can get good tonnage of ethanol. Sugar beet is used not more in our country, but in some countries like Brazil or somewhere else it is used. Some countries in the world they use it because it removes the step of dilution. See, for both sugar corn waste and sugar beet, you have got around 15% to 18% sugar content, which is has to be maintained for actual this one. So you get rid of one step. You can have grains. You can have grains like Jerusalem artichokes. You have abundant sources of ethanol fermentable material. That is a good advantage. But it has to be crushed to pulp and cooked for 2 3 hours. Dilution is not necessary. Basically, for all these 2 3 slides, you will see that dilution is not necessary because their natural sugar content is close to the required one. Just you have to adjust the pH and carry out the fermentation. Again, with potato is the same thing, but the sweet potato you have got more fermentable sugar, hence the dilution is required over here. For alcohol fermentation, generally batch fermentation is employed, having fermenter volume of 600 meter cube. So these are the standard parameters followed in the trade generally for actual production scale, not in the lab scale. So it is a long-term process first because glucose has to be produced, and produced glucose has to be utilized into pyruvate, and anaerobically it produces alcohol. So the first step in these 12 days, first 3 to 4 days, we are just allowing the yeast or inoculum to grow, and after growth it undergoes anaerobic fermentation to produce ethanol. So these are the maximum productivity guides. So 1 hour you will get 2 grams of ethanol around. So that's why you need more days to have a comparable higher yield. You can you can have a variation in this. If you look at this point, second last point, you can have a variation which is called as fed batch culture, in which you can reuse the cells which are used, and you can bring back again to the fermenter for next lot of inoculum and substrate. And it depends on the for our country, it depends on the quality of molasses. The max yield you can get is 95%, which is quite impressive. As you will see, continuous fermentation is not generally used here as a production of ethanol, because the yields are low and expenses to resources are high. Generally, you with some alcoholic beverage for fermentations, you may see continuous fermentation like beer or something like that. But mostly over there also you will get batch fermentations. So this is the diagram. Whatever we have seen: grinding, cooking, liquefaction, saccharification. So this is nothing but all these to remove the barriers which are there in between starch and microorganisms. So grinding and cooking remove the seed cover and all those things. Liquefaction and saccharification will convert that starch into oligomers. That's what biological process happening behind this physical process. In this particularly biochemical or microbial process fermentation, you will get primary product as CO2 and ethanol. CO2 as a byproduct, it is so uh, scarcely produced. It is released mostly in atmosphere. You can't pack it and pressurize it in the cylinder. But the ethanol is used, as we have seen, and distillation you will give you ethanol. But to what to distill, that involves the process recovery, ethanol recovery by centrifugation, evaporation. Then you have distillers grains, and distillers grains with solubles. These are the products. After ethanol recovery, whatever remains is this. So, okay, we have carried out fermentation and all that. Now we know that there is ethanol in the system, but how to recover that? It's mixed with everything. Leftover biomass, leftover substrate, which is still in minute quantities of starch and iron like that. We need to recover the ethanol. So that depends on end product metabolite, where it is located. If in general, product recovery focuses where your material of interest is in intracellular or extracellular. So for us, it is extracellular, it is in the medium. So we are not going to use cells, we are going to collect the cells separately and the medium, liquid medium, separately for alcohol recovery. So cells, as we have seen, it can be reused here. So in this separator, you can reuse those cells in fermenter by removing them from the liquid medium, and then how to separate those cells from the medium. You can have a very big centrifuge to separate the cell biomass. So these are the less frequently followed step, but the centrifugation step is the least followed due to the scale associated with it. So flocculation is the first step, in which a flocculation agent which will allow this process is. uh, so what is the flocculation? It produces large cakes of biomass with it's any agent which will bring about two cells of ethanol producing microorganism together, removing the solvent or the liquid layer in between. So it produces large aggregates which will still settle more readily than single cells. Other than that, you can have a flotation by introducing gas into liquid. Just bubble CO2 or any other suitable gas, except oxygen, of course. You can bubble any inert gas, nitrogen or helium or like that. You can have the flotation. The cells get trapped into the gas bubbles, and as bubbles are lighter than liquid, they will move to the top of the vessel. And on the top, you can have the cells along with that froth. You just collect the froth along with the cells and get removed from bioreactor. Other part you can have it as a filtration of bacterial cultures or filamentous fungi. You can have depth filters or static and closed flow filtration like this. Distillation involves separation of mixture of liquids. You know the distillation process quite a bit, so we are going to move fast. Low boiling point, solvents will vaporize first. The vapor passes into distillating head and condenser. What within a condenser, vapor is cooled and it liquefies. So here in ethanol fermentation, you will have this in the distillating pot. You will have more H2O in liquid. Most of the thing ethanol due to low boiling point will be there in the head space, which will go ethanol vapors will go to distilling head and condenser. So that's what I was talking about the pot still. Why pot still is used? We have seen this in one slide. That is have an advantage over conventional distillation like this. Fine flour of the raw material used is difficult to remove in distillation process. So it is a particulate matter of raw material, whatever starch source you are using. Uh, it may be sugar cane, sugar beet, or like that. Fine particulate matter creates a problem apart from cells. So you can't use conventional distillation, what you do in your labs, to go for this. Plus the scale is quite high. So pot still offers a convenient way to do that distillation in this manner. You have got a hard container provided with heating element. It will heat the container and the mash, and the vapor will contain your ethanol and water. It will get cooled in a cold water. So this is a boiling chamber, this is a cooling chamber, this is a water jacket and condenser. So drainage, cold water is in and drain. It is becoming due to water jack due to this contact of this hot spiral pipe in which where steam with vaporized ethanol is there. It gets heat exchange occurs and this warm liquid comes out to comes out from drainage. Again, you can recool it and provide it in the form of cold water or just treat this into an effluent treatment and just get it rid of. Like we have seen, ethanol has a boiling point of 75, 8.5, and water boiling point is 100 degrees. So you can use this difference in their boiling points for distillation. If you want very high grade ethanol, you can produce purify it using GLC or FPLC, particularly preparative grade. So the column size and diameters will be very huge. So these are the applications of ethanol apart from biofuel. We are not going to detail with this. So what we have looked into right now is this process, which we majorly skipped in last lecture, which is a product recovery. What we have seen in product recovery, there are separation of cell biomass, separation of fine particles of uh, raw material or starchy substance used using pot steel, and there are different separation of cell biomass methods: flocculation, flotation, filtration, and centrifugation. Most important methods are flocculation and flotation. In flocculation, you add flocculating agent so that it will combine all cells together to form large clumps, which will do under the influence of gravity. They will settle at the bottom of the pump, bottom of the reactor, and it can be removed. Otherwise, you can move all the cells into up by introducing gas into the liquid by flotation. So we have discussed already advantages, disadvantages in last lecture. I'm going to skip this biodiesel part. We have already discussed this. So coming to the next part, which is remaining from module 3, is biogas. So biogas is nothing but a gas produced by breakdown of organic matter in the absence of oxygen. So remember, in bioethanol also, in biogas also, you have to have absence of oxygen. That's why there are no aerators and there is no open lid or open system. It's always closed and under pressurized system. So reactor also, for alcohol fermentation reactors, they will not have any aerators, but they will have agitators. That means mixing of liquid contents is allowed, but not aeration, not introduction of air. So like bioethanol, it's still a renewable energy source like solar and wind. It can be produced from sugarcane waste, maize silage like that. But it is mostly produced by green waste, municipal waste, sewage, manure, such things. So depending on the waste, you can have either biogas, you can have either bioethanol or biodiesel, right? So biogas, why it is important? Because it has certain greenhouse gases. One climate change or greenhouse gases point of view, you are using these two main greenhouse gases, CO2 and CH4, methane and carbon dioxide, for a good purpose. They are getting burnt as a fuel instead of getting released in the environment. You can use those gases, and plus you have less emissions of methane and CO2 from animal sewage and manual sewage and waste, fecal matter waste. So you are good, you're getting something good out of the waste. Gases methane, hydrogen, and CO can be combusted or oxidized with oxygen. This energy release allows biogas to be useful. So when it is combined with oxygen, carbon monoxide, or hydrogen, along with that, methane is a fuel. Oxidizer or oxygen is provided by these oxidizers, carbon monoxide or like that, and it can be used. It allows it their use as fuel. Plus, biogas can be also clean. That means removal of CO2, because CO2 is not a good fuel. It is actually suppresses the fire. So it can be cleaned to have more amount of methane and carbon monoxide or like that. Hydrogen, particularly hydrogen and methane, to natural gas standards. Then it becomes biomethane and can be used as a similar to our CNG and natural gas. It can be pressurized also then. Here, conventionally, it is produced in anaerobic digesters, and during this process, as airtight tank transform the biomass waste into methane, it produces renewable energy. So there are two designs which are generally used for biogas. It is a floating drum in which you have a mixing tank, inlet pipe, digester. Whatever biogas or is produced, component gases of the biomass, CO2 and methane and all those things are get trapped inside it. As you go on adding the inlet and as it undergoes digestion, gases accumulate in this, and this is a floating drum. This gas holder is floating over this, and as the gases accumulate it will rise above the soil. So this is the soil level. So you will know automatically, there is no such any other indication, there is just a gas control valve. How we are going to know that the gas is getting produced? By looking at the gas holder. After some days or a few days, the as the biogas gets accumulates, it will shift under pressure. It will apply pressure to this gas holder, and this gas holder drum will rise its level. So increase in the level of this upper gas holder will tell you that biogas is getting produced. So from the head you can have a outlet for usage of that biogas. Despite of good design, it has got certain disadvantages. Advantages are higher gas production. Floating drum has welded braces. There is no problem of gas leakage, and most important advantage is you get a constant gas pressure, which can be used, and it is essential as a fuel. There is variable pressure, so slow pressure, your flame will go off, and it is of no use. But on the poor side, disadvantage side, it has got higher cost because it uses construction of those floating drums using steel and cement, and it has to be accurately uh, constructed using welded braces and like that. Another thing is if the gas holder is metal, you can see the heat loss. So whatever digester in the digester, whatever heat is produced, the gas is pretty warm due to microbial action, and it is getting lost. Gas holder requires painting once in a year. Okay, these are maintenance issues. Flexible pipe joining to gas holder to maintain. So it is a high maintenance stuff. For our country like India, there is an organization called AFPRO, which developed this particular model, which is called as Green Bandhu biogas model, which has certain advantages more suitable for our country. Like it has low cost, non-corrosion, it uses only steam, no steel. Heat insulation is better because you are using the cement also, there is no steel and everything is below the ground. If you look at this, all things are below the ground. Include there is no floating dome or like that, there is just a cavity which is used. And there is initial study that is the time everything is over here. There is no floating drum. Whatever headspace you have obtained, you will get a gas from that. Temperature will be constant. Cattle and human excreta long fiber stocks can be fed. So you can feed anything without any pre-processing. That's the advantage. You have got ACC pipe here mixing tank. Outlet tank entirely will be below the soil. So whatever manure you have, you it directly mix into the soil. So there is no need for any collection of waste coming out of this. But disadvantage is you have to rely on masons and craftsmen for in the rural areas you may not find those, but that is not particular problem for our country where we have good population of workers also along with the general population. But the main disadvantage is gas production is less and it has a variable gas pressure. There is no controlling gas holding mechanism is not there in this. Hence you will get a variable gas pressure and there is no stirring arrangement. So it will form a scum. So here if you know don't stir this, all solids will get settled and this area will decrease and it will lose the efficiency of that system. That is called as scum. So you have to partially time to time you have to clean the thing. And as long fibrous stalks if you feed, it will require more and more cleaning. So it has got mixing tank, pipe, feeder, inlet, digester, tank holders, slurry outlet, gas outlet, and all these components. In anaerobic digester, it is a series of processes in which microorganisms. So we are looking into microbial processes behind this biogas production. It, which are the anaerobic processes in which microorganisms you use to produce biogas. So the digestion process has got certain steps. First, bacterial hydrolysis to break down insoluble organic polymers, followed by acidogenic bacteria to convert to which bring the pH down to acidic side of those mixture, and they produce ammonia, CO2, hydrogen, organic acids like acetic acid and acetogenic bacteria also along with this to convert these organic acids into acetic acid. So these organic acids are simple acids like HCl, which are further acted upon by acetogenic bacteria, which will mainly produce acetic acid along with ammonia, hydrogen, CO2. And finally, it will get acted on by methanogens, which convert their product to methane and CO2. Like we have seen, there are three steps: hydrolysis, adhesive formation, and methane formation. So in hydrolysis, fat, starches, and proteins are broken down into simple compounds, similar to your alcohol production. It is also called as polymer breakdown stage. End product is glucose. Very similar to alcohol production, just your raw material is waste, sewage, and fecal matter like that. The result product from hydrolysis serves as an input for acid forming bacteria. They will produce acidic acid, propionic acid, butyric acid. So they will acidify the medium, followed by methanogens, which will produce this acidic acid produced in earlier steps to convert them into methane and CO2. So these are obligatory anaerobic, very sensitivity to environmental changes. So this stage is more critical. It utilizes intermediate products and converts them to methane, CO2, and water. Plus, methanogens are sensitive to high and low pHs, and it occurs only between 6.5 to 8. So if you have got higher acid production and less methane formation, it is of no use. So these are three stages: fermentative, similar to bioethanol, protein carbohydrate fats broken down to simple products. Stage 2, acetogenic, where acids, alcohols, and acetic acids are converted to acetic acids. And in third stage, methanogenic bacteria use acetic acid to produce methane. Same thing. So these are the reactions involved. These acid methanogens use this acetic acid to produce methane and CO2. In ethanol fermentation, you have this in oh sorry, not in ethanol fermentation. Here, whatever alcohol you get by anaerobic state, it is getting mixed with CO2 again by methanogenic bacteria, which will produce methane and acetic acid in the second step, acetic acid formation, acetogenesis. Some bacteria combine hydrogen and CO2 to produce methane and water. So in this, all these reactions, whatever methane is produced, which is a chief component of biogas, along with CO2, it is used as a fuel. Generally, you will see that methane is 50%, CO2 is 25%. NO as a breakdown product of amino acids, nitrogen is here, nitrogen oxide, 10%, and hydrogen is very less, around 1% or so. So it is used for various uses, using in electricity generation from gas for burning purposes and engines like this cooking, heating, lamps, mechanical energy, like this. What is the advantage of biodiesel? Biogas is the slurry which you get after digestion, as which gets out to this right hand side, this outlet tank, whether it is a bean bamboo plant or floating drum plant. The slurry or the overflow can be used as a manure for fields. Why? Because it is odorless. If it is odorless, that means it is incomplete. So all types of manure, not only with the biogas, for other manures also. Whenever you see that the manure is having odor, you throw away that manure. That manure is not a manure. This is undergoing still process of fermentation. And all manures are odorless when fully digested, like this. It, as it is odorless and everything is utilized, naturally it will not attract any insect or flies in an open condition. Apart from that, surprisingly, it will repel termites because it has no starch and this thing, but it has some other components which are toxic and their pH has been varied. So it will repel termites. And raw dung, if you can dry this product out and by removing the water or solvent part in it, and you can use it as a farmer yard manure. But if the dung is raw, that means your manure from the sewage from animal and human waste attracts insects and flies, and they can harm plants fertilized with farmland. So when it is completely digested, it is safe and good to go. Effluent used as a fertilizer, it reduces weed growth. So these are again added advantage because due to their chemical contents, effluent can be reduced for controlling weeds also. Now coming to the last part, why don't we see biogas in vehicles? If a biogas has to be used in power vehicles, there are certain barriers in that. First step is we have to get rid of CO2 because the presence of CO2 lowers the power output from the engine. Plus, it takes space in the storage cylinders. Thus, they reduce the range of the vehicle, and it can cause the problems of freezing at walls and metering devices. So CO2 has to be removed from biogas, and then you have to pressurize the biogas to be packed as a cylinder, which increases the cost dramatically. So in a cost benefit point of view, it is not good to have biogas in vehicles. So coming to the end of this module, uh, we have almost covered the module 2 in this lecture, and next time onwards, next the thing, only one thing which is remaining in module 3, it is nothing but production of hydrogen gas. So that we will be covered in the another video. Till then, we have covered all these points from 3.1 to 3.4, including biodiesel from hydrocarbons. So in the next video, we will cover the microbial production of hydrogen gas, one point, and then we will declare this module as a completed. That's it. Thank you for patiently listening.